Prosserman. He is a first-year graduate student at Queen's University. He completed this project for his fourth-year undergraduate honor thesis with Dr. Janet Menars. He hoped to shed some light on the importance of understanding how individual ventilated cage system can impact lab rodents and their behavior. So I'm really looking forward to your talk, Alex. Take it away.
Thank you. So can you hear me? Perfect. Okay, so hi everyone. My name is Alex Prosserman, and I'm a first-year grad student studying neuroscience at Queen's University in Kingston, Canada. So today I'll be talking about the influence of social housing and air pressure on rats' anxiety-related behavior, similar to Emma. So to start, most federal committees concerned with the regulation of animal care are mandating a principle of refinement, which is changing some aspect of an experiment that results in a reduction or replacement of animals, or a reduction of any pain, stress, or distress that animals may experience. One of the most recent refinements is the replacement of classical open-top cages, shown here on the left, with new individually ventilated cages, which I'll now refer to as IVCs, shown here on the right. IVCs are sealed cages where air must pass through a filtering system before entering the cage. And the cages have two air ventilators to control the air pressure, one pushing air into the cage and another pulling air out of the cage. The cages can be ventilated under either positive or negative air pressure, and this can be a kind of a confusing topic, so I'm going to walk you through it. Under positive air pressure, there's more air being ventilated into the cage than is being ventilated out. So as you can see, there's a higher concentration of air inside the cage than outside the cage. Because of this, the air inside the cage passively diffuses out in an attempt to balance this concentration gradient. As all of the air entering the cage is filtered, rodents can be protected from airborne diseases. However, a drawback of positive pressure is that the air may dry out the cage environment, which has caused aggression in rats. Conversely, under negative air pressure, more air is being pulled out of the cage than is being pushed in. So therefore, in an attempt to balance the concentration gradient, some air passively diffuses back into the cage. And as all of the air leaving the cage is filtered, this can protect lab staff from airborne allergens within the cage. IVCs are also beneficial because frequent air changes improve its microclimate by eliminating the buildup of carbon dioxide and ammonia. However, there are some drawbacks to using IVCs. Firstly, some evidence suggests that rodents living in IVCs can consume food in a typical manner. Secondly, IVCs can alter behavioral phenotypes, such as a well-characterized genetic model of schizophrenia that was completely altered when switching from OTCs to IVCs. And finally, and most importantly for my study, some evidence suggests that IVCs can increase rats' anxiety levels. So because IVCs might increase rats' anxiety, we thought maybe we can alleviate that by pair housing them, because it's common for rats housed in pairs to be less anxious than rats housed alone. So therefore, our primary question was whether single and socially housed rats living in IVCs would differ in their responses to an anxiety test. And also a supplemental goal of our study was to determine if rodent behavior could be influenced by maintaining the IVCs under either positive or negative air pressure. Social housing may also have implications on the body's oxytocin system. Oxytocin is a hormone produced by cells in the paraventricular nucleus and the supraoptic nucleus of the hypothalamus. Oxytocin has anxiety-reducing effects on rats. For example, oxytocin administration decreased rats' corticosterone response to noise stress. So this brings me to my third question. Could social housing in IVCs also influence the density of oxytocin-producing cells in the hypothalamus? So to answer these questions, I used 40 male Long Evans rats from Charles River, Quebec. They lived on a 12-hour light and dark cycle with free access to food and water. And when they arrived, 10 rats were randomly assigned to each of the following 4 conditions. The first was single housing and positive air pressure. The second was single housing, negative air pressure. The third was paired housing and positive air pressure. And then the last was paired housing and negative air pressure. So after the rats lived in their respective housing conditions for 6 weeks, I conducted the Novelty-Induced Suppression of Feeding test, also called the NSFT. This test compares a rat's latency to consume a tasty snack in a familiar and novel environment. So what I did is for 4 consecutive habituation days in a row, The rats were placed in a clear open top cage with a bowl containing a graham cracker, shown here by the picture on the left. Then I measured the time it took for each rat to initiate the consumption of the graham cracker. And if they didn't begin eating after 10 minutes, I ended the trial and placed each rat and the graham cracker back in their home cage. Then on the 5th day, I conducted the same test in a novel room with a novel cage and bowl, shown here by the picture on the right. And prolonged times to begin feeding were an indication of anxiety. So before I conducted the test, I predicted that single housing would increase the rats' latencies to initiate snack consumption in the NSFT, suggesting increased anxiety-like behavior. I also predicted that positive pressure would increase their latencies, because as I mentioned, positive pressure can dry out the cage environment, which has caused aggression before. So what did I find? Well, my first result illustrates that there was no effect of social housing. So here on the y-axis is the rat's latency to initiate snack consumption measured in seconds. And on the x-axis was the testing day with habituation days 1, 2, 3, and 4, and then the novel cage test on the 5th day. And as you can see, there is no difference in the rat's latencies to initiate snack consumption if they were pair-housed, shown here by the solid line, or if they were single-housed, shown here by the dotted line. And honestly, this result completely surprised me, as the anxiety-reducing effects of social housing are quite robust in the literature. So my first thought was, could these results be due to an error in the testing procedure? But as you can see here, I obtained a characteristic U-shaped latency profile of the NSFT test, whereby the highest latencies occurred on days when novelty was a component. That is on the first habituation day and during the novel cage test day. And this was irrespective of housing and air pressure conditions. So this suggested that my failure to observe evidence of social buffering does not appear due to problems with the testing procedure, and that IVCs may be inherently stressful for rats. So next, and counter to my original hypothesis, I observed a marked main effect of air pressure, and that is rats housed under negative air pressure, as indicated by the solid line, took significantly longer than those housed under positive air pressure, as indicated by the dotted line, to initiate snack consumption. And this implies that the rats living under negative air pressure were more anxious than the rats living under positive air pressure. So importantly, there were no group differences in body weight, which suggests that appetitive factors did not contribute to these group differences. One possibility is that the rats housed under negative air pressure perceived a relatively greater threat from the possibility of facing conspecifics in the colony, that is, other rats in the colony. So under negative air pressure, the rats were exposed to cues from the colony room, such as the sounds and scents of other rodents. It follows that the rats living under negative air pressure might have been more aware of the presence of other rats in the colony room. And since rodents naturally compete for food sources, the rats living under negative air pressure may have suspected that they were entering the territory of other rodents during testing, and thus exercised greater caution to initiate snack consumption. Conversely, the rats living under positive air pressure were not exposed to olfactory cues in the colony room, which might explain why they were less cautious during the test. But ultimately, it's not really clear why the rats living under negative air pressure took longer to initiate consumption in the NSFT. So onto the immunohistochemistry component. So to count the number of oxytocin producing cells in the hypothalamus, I used antibodies that specifically binded to and labeled those cells. Then I used a microscope to count the number of immunoreactive cells. And I predicted that single-housed rats would have fewer oxytocin-labeled cells in the hypothalamus. And my immunohistochemical analysis revealed a marginally significant housing and air pressure interaction, such that single-housed rats housed under negative air pressure had significantly fewer oxytocin-labeled cells in the posterior parvocellular region of the paraventricular nucleus relative to single-housed rats under positive air pressure. And given the purported role of oxytocin in anxiety reduction, it seems likely that single-housed rats under negative air pressure were more anxious than single-housed rats under positive air pressure. And this outcome maps onto my main effect of air pressure in the NSFT, but it's not clear why this was only apparent in the single-housed group. And if anyone has any ideas or comments, I'd love to hear from you about that. So to summarize, firstly, there is no evidence that social housing influenced the rats' latencies to initiate snack consumption across the habituation or test trials in the NSFT. Secondly, there is a marked and unexpected main effect of air pressure, such that the rats housed under negative air pressure took longer to bite the palatable snack than those housed under positive air pressure. Finally, single-housed rats under positive air pressure displayed more oxytocin-labeled cells in the PaPo of the hypothalamus. And these results suggest that IVCs can alter the well-characterized anxiety-reducing effect of social housing in laboratory rodents, and actually might in fact influence other well-characterized displays of rodent behavior. So it's really important that we further comprehend how individually ventilated cage systems can impact laboratory rodents and their behaviors. And this would both to ensure that research efforts are robust and to advance the well-being of animals used in research. And at the very least, my findings suggest that researchers need to cautiously consider the form of housing condition used for experimental rodents. So to finish up, I'd like to sincerely thank my supervisor, Dr. Janet Menard, as well as NSERC for funding our research, and Samantha Irwin and Emma Littler for their support and help with this research. So thank you.
Thank you so much. And let's see if there are questions. While we're waiting for questions, I will ask something. So I didn't quite get it that how the negative one, what was your explanation for when the negative pressure will make them feel like more competitive for food?
Because under negative air pressure, they're exposed to sounds and scents from the colony room. So my explanation for that is they might have suspected during testing that they were entering the territory of other rodents and therefore exercise greater caution to initiate snack consumption, whereas the positive pressure rats weren't exposed to these cues. So during testing, they would possibly be less cautious.
So we have a question. Wonderful talk. One question, is this phenotype related to IVC specific to rats or can be seen in mice as well?
In the study, we just used rats. I'm actually not too sure if it can be seen in mice as well, but to the best of my knowledge, it is only rats.
Very interesting. I think I even have more questions. Yeah, do you think it is the nature of the task that it is sort of, because social housing give them the sense that there is a possible competitor for the food? Do you think that might be weakening the benefits of social housing because the behavioral test is very food related and it introduces a sense of competition?
Yeah, definitely. That's a really good point. I didn't think about that one before, but yeah, definitely that could have an effect.
That's really good. I want a tasty treat, but
yeah.
And great. Now I think we should move on to our next